Just briefly, I wanted to review where we last left off. We were talking about the fluid mosaic model and the fact that not only does this mean that there's fluidity to the lipids in the membranes, and that's based off of their composition, what type of lipids that there are in each membrane, and it can change from tissue to tissue, organelle membrane to cell membrane, and between the different organelles, and also even within the leaflets of the same membrane itself. And so the example that they usually talk about is the cell membrane. And we'll talk about one specific example and the importance of it later on, most likely today. And then I can't, since it's no longer in PowerPoint form, I can't click on it to show that table, so I just went ahead and I put it, if I can bring it up. So it's right here. And so this was, and I wanted you to be able to explain the rationale about the fact that between the different membranes here, for example, why that they have such different compositions. And so if you just looked in particular about the mitochondrial inner membrane, which is the one that people had the most problems in discussing, was why is it so proteinaceous? Well, why does that help its functionality? What is it about it that makes it need to have such a high protein content? What does it do? It's the electron transport chain, and it does also do ATP synthesis, and ATP synthesis itself occurs in an enzyme complex. Some people call it complex 5 of the ETC, but technically it is not a part of the electron transport chain. It's part of the oxidative phosphorylation. And the two processes are coupled. But so the ATP synthase is also proteinaceous, and then the electron transport chain itself. Okay, and technically speaking, one protein of now my mind's slipping. Citric acid cycle. I knew it'd come to me. Citric acid cycle has one complex that does associate with the inner membrane as well, of the mitochondria. And we'll go in more detail with that with respect to next semester. And then we also discussed, for example, the reason why that there's so much lipids in myelin, and also the higher carbohydrate concentrations in blood cells. And I wanted you to be able to talk about the logic behind the reasons why that would make sense. Like, why don't you want lots of proteins in the myelin, and what the whole role of the myelin is, and so on and so forth. You have to do that every single time. It doesn't advance on its own. Okay, and this is just a figure from your book. Technically, I think it's from 1 or 2 editions back, but it just shows you the different types of proteins and lipids that can occur within just an average membrane. And so we have, for example, so we have integral versus peripheral proteins. And so what does peripheral mean?
Periphery.
It's on the periphery. And so these are the ones that are not, sometimes they may have an anchor, like a single lipid or two that will anchor it in there, or many times it's held by interactions between the membrane and the protein itself, but they are not, the protein itself is not inserted into the membrane. It does not span the membrane. And so do you think it's going to be relatively easy or difficult to separate the peripheral proteins from the membrane? It's usually easy, okay? And we'll talk a little bit about that in a moment. Versus the integral proteins. And integral proteins are ones that do insert themselves into the membrane. They could span the membrane, and we're going to talk about different types of integral proteins in a moment, where sometimes they do it just once, sometimes they span it multiple times. They may have, parts of the protein exposed on, one side or the other, or even on both sides. Okay, and then glycoproteins is just, so an example would be your antigens that have long sugar chains, polysaccharides that are exposed on the outside, typically, of the cell. You can have glycolipids, which we've talked about, for example, with some of the galactosides and things like that. And then we have the different types of lipids themselves, and we've already discussed the phospholipids, sphingolipids, The sterols like cholesterol, cholesterol esters, so on and so forth. But what kind of interactions do you suppose are going to occur, say, between the peripheral protein and the membrane? If we look at this one, this is a, looks like I'm trying to see the colors, but this is a good example of the peripheral protein. What kind of interactions is going to hold that in place? Is it going to be hydrophobic? If it has a lipid anchor, for example, like the RAS protein that gets farnesylated, can have a hydrophobic interaction because it's got that literally a lipid anchor that holds it in place. But that's exactly it. So the head, the phospholipid bilayer's head tends to be hydrophilic, right? Because it's aqueous, and so many times it's going to have phosphate. Typically, it's going to have at least a negative charge. So that can be an ionic interaction, actually. So it really depends on if it's anchored. Like this would be an example of like the farnesyl group or the geranyl group that's holding it in place. And so the one that we've talked about in the past was RAS, and in chemistry of cancer we've gone in a lot of detail with RAS. Next semester we'll go into a lot of detail with RAS too. Or you can have it to where it's an ionic interaction. And so if it's held by ionic interactions and we're doing some type of protein purification, like for example next year in lab, if we wanted to purify peripheral proteins, you can many times just compete them off with a detergent, with some type of salt, right? Because if it's an ion-ion interaction, you can break it apart with salt and it'll come off. Whereas in integral protein, what kind of interactions are going to be occurring within the membrane? Like right here, for example, in this alpha helix, what kind of interactions would be occurring between the alpha helix and the membrane itself? That's going to be hydrophobic. And so if you were trying to purify and get that protein out to characterize it, what would you have to do to it? If you add salt, it's going to make it even worse, right? Because it's going to want to exclude water even more. So what do you add instead of adding salt? You do this all the time. Most of us do it pretty much every morning. Right, soap, detergent, right? Because you want to do something that's going to be amphipathic, has a hydrophobic region and a hydrophilic region. So, or if you do your laundry, you're adding in detergent. It's the exact same principle. Because the hydrophobic portions of the soap or the detergent will protect the alpha helix, whereas the polar head groups will be fine with the water and it just washes on out. And so that's why an integral protein does require some type of detergent in order to get it out of the membrane. Otherwise, it's going to stay in there. So they're usually held quite tightly. Okay, and of course you can have channels. Channels are just a specific type of integral proteins. And so the composition can vary within the cell. This is just another way of showing what I showed on that one website in the table form. This is just a bar graph. And once again, I would expect you to be able to discuss a little bit behind the logic on some of the ones that make more sense than, I shouldn't say make more sense than others, but those that do make sense. So for example, once again, if we compare the differences between, let me see if I can find it, the plasma membrane versus, where is it? For example, either the nuclear membrane or even the ERs. Like the big difference there has to do with cholesterol. So what will having extra cholesterol do to the plasma membrane that it doesn't do to, say, the nucleus? Why do we want more cholesterol in the plasma? It's going to make it more rigid because you just don't want all of the stuff leaking out or the stuff to come in too easily, either come in or come out too easily. Whereas what's going to happen with the nucleus itself? What occurs between the nucleus and the cytosol? What are some of the examples? I mean, there's lots of things. The nucleus is like that. Well, I don't call it the Holy of Holies. You're going to hear me refer to the Holy of Holies a lot as being the matrix of the mitochondria. Because you're going to find out it's very difficult to get inside the mitochondria. But what happens either in the nucleus that has to come out or stuff that's in the cytosol has to come in? Yeah, you're going to have RNA and so you got the nucleic acids shuttling in and out. You're going to have proteins that are going to come in and out. And so you've got a lot more action and exchange that needs to be occurring. So it's much more fluid than that for the plasma membrane. Okay. So here I've actually already discussed a lot of this, but this is putting it into text form. The fact that the membrane proteins can have multiple functions and functionalities. I think you already know that from cell and molecular biology and from genetics and general biology. Some of the examples are that they are receptors. So for those of you who are taking immunology, you probably talked about the epitope versus the epitope versus the antibody this reaction. I cannot speak today. Or even like the integrins, which we'll discuss in greater detail in chapter 12, I think it is, which is for cell recognition. You could also have them be transporters. So what's an example of something that gets transported across the cell membrane? Anything? Calcium ions, right? And so calcium for like muscle contractions. You also have calcium for nerve impulses, or sodium and potassium and all the ions. And you're going to find out there are certain things that that do not cross the average generic cell membrane. For example, like insulin. Insulin never comes across the membrane on its effector cells. Okay, instead, what it's going to do is going to bind to an integral protein because that's the receptor site. And you can also have enzymes that do their catalysis right at the membrane. Okay, I have to...
What does that mean when people generally denatures come?
Oh, I'll explain that in just a moment. I was going to try to, because I don't know how, I can see it easily, but I don't know how well you guys can see it down there. All right, so we've talked about the peripheral proteins many times. It's electrostatic interactions. It could have an anchor, whereas like the farnesyl group or the general group, it's a lipid that's added onto it. Usually you can remove it as long as it's not anchored too well. You can remove it just by increasing the ionic strength. And that's just a fancy way of saying that you add in lots of salt and the salt will compete with the binding. But the integral proteins, you have to use a detergent, okay? And if you use the right kind of detergent, you can keep the structure fine. However, many times, if you think about it, and I don't know that I'd be able to do this justice, but if you have some type of, let's say, an alpha helix that's hydrophobic, oops, I've got more room to write on my computer than on there. Whenever you take that out and you're trying to pull it out of the membrane, at some point in time, if it's exposed to water, it's going to denature. It's going to get a different shape or a different structure. That's what they mean by that. So you have to be very careful. And the ultrasonification is where you use sound waves. And so we can do that now in the lab. And so it literally puts sound waves, like it's got a little probe on it, you stick it in there, and it reverberates at a really high frequency, and it causes the cell membranes to break open. That many times will definitely denature. Unless you put enough detergents in there, it's going to denature those proteins because of the fact that those proteins will be exposed to some water. And so they'll either clump up together to form some type of agglutinate mess, a glop, or it's going to want to fold in on itself and have the incorrect fold. Okay. And then you can have something that's amphitropic, which means it can be found as both. Okay, it's reversible binding. Okay. And so that's the one where I've been talking about, like the farnesylation. That's just the lipids. Palmitoylation. That's just when they add a palmitic. It's a fatty acid that can be added on there. Okay, and so those many times can be like a regulatory protein. So the example that I've talked about before with RAS doesn't associate with the membrane unless it's farnesylated. And so if it doesn't get farnesylated, it can't be active. Okay, and if it can't be active, then you don't have to worry about it increasing transcription of cancer cells, assuming that you have mutant RAS. Okay, and we're going to talk a lot about that in chapter 12. There's a lot of the cascades and signaling pathways are due to some type of modification of a protein. And many of them, all except for one class, happens at the membrane. So you have some type of external signal trying to create an internal effect. Ooh, that sounded really good. I got to try to remember that for next chapter. External signal. Where you have an external signal trying to create an internal effect. Yeah, got to remember that one.
I had lame jokes.
Oh yeah, you should have heard the talk last night that we had, because it was with Dr. Davis was the speaker at the board dinner.
Oh yeah, he has very lame jokes.
And so he get talking about his puns. We had lots of puns. So yeah, they were. Some of them were.
I told a joke about the bear, and like the one where the punchline is like I don't have to be able to outrun the bear, I just have to outrun you. And he's like, I'm sorry, that was Grizzly.
yeah, there's those kind that he would. Yes, with the discussion on determinism versus propensity with respect to gender identity issues and sexuality. Light dinner topic. It was a board dinner, and so yeah, light dinner topic.
Yeah.
Okay, so this is just a cartoon depicting what I've been saying verbally, and so here we have. Let me switch it so I don't drop. Just a peripheral protein that's actually, this one's being held between protein-protein interactions. And so they're just showing here for calcium, for example, being able to compete it off. Where it says like urea is actually a denaturant, and so that's something different. Chelating means it can take away all the ionic, like the metal ions in particular. And carbonate, of course, is an anion. But we just have some type of ionic interaction that you're able to compete off. If it's, I'm going to come here first, the integral protein, whereas, what is that? The lighter color there is to show the hydrophobic interactions, and so they've added in some type of detergent that where essentially the detergent is competing with the lipids here for access to the hydrophobic part. And so if you do it just right and using a gentle detergent, then you're able to get the integral protein out. And so for years and years and years, it was difficult, almost impossible for them to crystallize the hydrophobic proteins and proteins for lipids because that are in your membrane. They would only be able to crystallize the portions that were cytosolic or external, because whenever you make a crystal, it's in an aqueous environment. Usually it can take like months a lot of times for a crystal to form, because you have to slowly let the solution that it's in dry, like evaporate, and then crystals are left behind. That's the most common method. And so they couldn't ever, they just had lots of difficulties with hydrophobic proteins. Then they started playing around with some different types of detergents, and they've now been able to do many hydrophobic proteins for crystal structures and for NMR. But that's why sometimes whenever you look at some of the hydrophobic proteins, crystal structures, they'll have like these weird things attached to them, and it's because it also crystallizes the lipid detergent that's alongside it, or the fats that are alongside it. But and then here's one where we have an anchor, and so literally if you're going to try to rescue that one, many times you just use the phospholipase or whatever lipase it is that can cleave it. Okay, and so that way you biologically can remove it. And then this is amphitropic, and so it's just showing you that how, depending upon what process it is, maybe it's biologically active whenever it's bound to the membrane, maybe it's biologically inactive when it's bound and it's not active until it gets released by the membrane. But it could be either way. So unfortunately, there's no other way except just to memorize these. There are different classes of integral proteins. Okay, and so there are 6 types. And what dictates makes them different, let me see if I can, there we go, is the directionality. Remember, proteins, we typically, when we talk about like the first amino acid versus the last, it goes from the N terminus to the C terminus. The number of times the protein goes across the membrane, and then the number of total polypeptides, like if you have a quaternary interaction between multiple proteins, and whether or not it's got a lipid anchor, okay, like the farnesyl group or the palmitoyl group, something like that. And so the only difference between type 1 and type 2, these are both proteins that only span it once. And it's whether or not the amino terminus is on the outside or the inside. So for type 1, the amino terminus is on the outside of the cell. Type 2, the amino terminus is on the inside of the cell. So that's the only difference, but it only can span at one time. And that's important because, well, the most obvious, hopefully, is the fact that they would have two different charges. If the amino terminus is on the outside of the cell, that's positively charged. If it's on the inside, that means the outside of the cell is showing a negative charge there. At a minimum, if not additional modifications. Whereas type 3 means that it spans multiple times. It's a single polypeptide, a single protein, but it goes up and down, up and down. Okay, and so many times I'll even talk about, okay, this is helix 1, helix 2, helix 3, depending upon, and many times it's alpha helical. They are not all alpha helical, but many times it is alpha helical. I'll show some pictures of some of the exceptions that are little pieces of sheet, so to speak. Actually, they're usually really big pieces of sheet because they make big barrels for like ion channels and things like that. Okay. And then type 4 is when you have multiple proteins that come together. Now, these are kind of interesting in the sense that if you think about it, let me see, I'm going to try to draw this. So I want to scooch that on over. So we have our membrane. I'm going to draw a couple. Except my phospholipids look more like something from a biology class. Okay. All right. And so let's say that we have this protein here with an alpha helix. We've already talked about like what kind of interactions usually occur between the alpha helix and the lipids in the membrane. What kind of interactions are occurring, say like between don't do that. Between this alpha helix and these lipids right there. What are going to be the interactions, usually? It's going to be hydrophobic. But let's say that we've got a type 4 kind of integral protein, technically be an integral protein complex, since it's two different proteins. So I'll do it in that color. Now, and it's true, it's still true that this could very easily be hydrophobic interactions here, but what about interactions between the two alpha helices? What kind of possibilities do we have then? Like what is that, red and green?
Yeah.
How does the red one and the green one know, hey, we should be partners in this sea of lipids? You know, it can be electrostatic, and sometimes that's very, very important, because let's say that the red one's got on one side is hydrophobic, but on the other side it's got some positive charges. And the green one side is hydrophobic, but it's also an amphipathic in the sense that the other side's got some negative charges. Well, the negative side and the positive side is going to come together. Or it could be hydrogen bonding, like polar interactions. And by doing that, they actually form a really strong bond because, you know, it's sort of like, it's not faith integration, but like romance or love integration. When the world is against you, the opposites attract, you can have each other's back. Sort of like that. I don't know. I'm reading the book of Job. But I'm not to the part where Job is being reaffirmed yet, so the world is still against him, so I can't integrate anything from the book of Job. He'd be like the red helix that's floating in the lipid sea right now. So, okay. And then finally, for type 5, what whoops, I shouldn't say finally, there's actually technically 2 more left, but here I'm going to bring this back up. Throw that one out. For type 5, that's one that has the anchor. Okay, so that would be like the RAS that got farnesylated. Something where it's got the palmitoyl group, a farnesyl group, a geranyl group, something that's hydrophobic has been attached to it and it's holding it in place. It's on its leash, so to speak. And then type 6 is where it's got more than one. You can follow, it's like a catch-all. And so the picture that they're showing here is, oh, yes, it's spanning at once, but it's also got the anchor in there, and so it's where it doesn't fit into one of the nice other categories. And once again, we're going to find, well, we've already talked about the fluid mosaic model, so it really does matter which one, it's got the right side exposed, and we're going to talk a little bit about the lipids and why that's important. Because if you get the wrong, it's literally the difference between life and death if it's the wrong thing that's on the outside versus the inside. We've actually been discussing this as I've gone along, but the things to consider with respect to like what amino acids comprise these membrane proteins. And I really wish I would have left just a blank figure to show that background before, where you can clearly see where you got the phospholipid head groups and the hydrophobic, inside. Because it's a lot better than my drawing. I know it's hard to believe. But in here, you're going to really want to start to think about what kind of properties the amino acids have versus the properties of the biological membrane. And remember, it's location, location, location. So, okay. So we've already talked about the fact that there's hydrophobic. So let's say, for example, that we've got this protein. Let's draw it right here. And so, whoops, it's got a kink in it, probably proline. But you got some type of protein right there. And so we've already said that here on the inside, we would have, assuming that we're just talking about one, either type 1 or type 2, where it's spanning once. What kind of amino acids would be right there, usually? Since it's going through the membrane right there. So Riley, what kind of amino acid? What's one of the possible amino acids that would be good to have inside that membrane?
It could be valine.
Like valine?
Isoleucine.
Isoleucine, leucine, glycine, proline. Any of those hydrophobic ones, because this is a very hydrophobic environment. Okay, so it's very hydrophobic, assuming that we're not talking about protein-protein interactions, but it's going to be some type of hydrophobic interaction to help hold it in place. Okay. However, what happens if, let's just, because I don't want to put in too many. What about right here? What kind of amino acids would be around the end, the way that I haven't drawn, right there at that part of the... Possibly cysteine, serine, okay, and why? What properties do they have that makes them good right there?
Hydrophilic.
They're hydrophilic, they're polar. What else? If it's a phospholipid, what other amino acids do you think that you'd expect to see? What does every phospholipid have? Every phospholipid have, besides a lipid?
Negative charge.
And it's going to have a negative charge, or it's going to have a phosphate. And so, what kind of amino acids, many times, do you expect could interact with a phosphate? Pardon? Someone said, I think.
I said positive ones.
The positive ones. And so what's some of the positive ones? Arginine. There's 2 more. Lysine, and the third one that's sometimes positive, sometimes not. Histidine. Mm-hmm. So I've got pictures that'll show you like how this is true. Okay. Then we have like down here, the ones that are now out either in the cytosol or the extracellular space, because I didn't say which is if that's inside or outside the cell. But regardless, it's an aqueous environment, so what kind of amino acids now will you see typically, or are more likely to see?
So hydrophilic.
The hydrophilic, the ones that are still polar. Typically, if you see hydrophobic ones right here, it's because they're making some type of important interaction to hold the protein to itself or something like that for folding reasons. Okay, now there's one little exception. I don't know, will this let me turn it? Oh, it will. Awesome. Is sometimes, and I will not be able to draw this, right at the interface. So you've got, say it's roughly smooth, you know, that sea of lipids. There's a type of interaction that can occur with the aromatics, where the aromatic ring will be a parallel right alongside it. And so you may see like a tryptophan or a tyrosine is really good because it's also got an alcohol group where it just lays like right on top of it, like it's almost floating. And so it's almost like a little cap. So once in a while you'll see an increase of just those aromatic ones that can form a special bond, because unfortunately we don't get to talk about it, but there are a couple different ways that aromatic compounds can bond. They can bond through like pi interactions where they stack on top of each other almost like a hamburger bun, and then they can also bond where they go in orthogonally, not perpendicular, where you have the hamburger bun with another one going in up and down, which I don't have a food analogy for that, I'm sorry. Sorry, but okay, advantage, and that's called so we're just going, the example here is glycophorin. And they use what's called the hydropathy plot. And the hydropathy index means how hydrophilic or hydrophobic something is. If it's a positive number, it means it's hydrophilic. If it's a negative number, it means it's hydrophobic. If it's 0, then it means it's completely neutral. And what they did is this one, for example, is you can, let's say that you've found out you're trying to isolate this protein, but you don't know, you know it's associated with the membrane because it requires some detergent for you to get it out, okay, to isolate it, but you don't know anything else about it. And so you sequence it, you degrade it, and you sequence it, you find out what the sequence is, and then based off of each amino acid, you plot how hydrophilic or hydrophobic it is. And what you notice is that you'll find out that it's really, really hydrophilic, and then we've got this one portion that's really hydrophobic. And so you can predict the minimum number of times it's inserted or it transverses the membrane. This one, you would say, oh, it most likely will only go in the membrane one time. Okay, it's going to have one major insertion in the membrane because it only can transverse it once. Okay. And they've color-coded it, but I can't tell all of my colors apart. But this one, they know the crystal structure. They actually know the crystal structure of glycophorin, but I forgot to copy and paste it from the PowerPoint because the PC version won't let me upload a PowerPoint into it. I have to do these individually as pictures. It's a pain. But that way we don't have to worry about Apple TV. But they've color-coded these, and so they've already crystallized it, and so you can see like how many times. But if you do its hydropathy plot, there it is. And so whenever it's once again it's color-coded, but you can see at least get an idea of the minimum number, even if you didn't know what the structure was. If you only knew the primary, remember the primary amino acid sequence is just literally from the N terminus to the C terminus, and they plot the residue number as a function of its hydrophobicity, essentially. And you can see, we're hydrophilic, hydrophobic, hydrophilic, hydrophobic, so on and so forth. And so you can see here that it's going to be a minimum of 6 to 7 times. And lo and behold, it really is once they crystallized it. In fact, this is one of the ways that one of the methods that they use for putative proteins to try to help them figure out what the crystals are and what kind of interactions that they should use in order to get better crystals or better NMR structures. It's a hydropathy plot. And there are free bioinformatics programs and apps out there to where if you have a protein sequence that you can put it in and it will do the hydropathy index for you. And so it'll just plot it out and it'll say, okay, this is what it looks like. And then you literally count the number of times. Oh, these are the hydrophobic regions. And so if we think it's a integral protein, then you can count the minimum numbers. And these are, so if you notice, for example, here, where 6 and 7 are, it doesn't completely come back. You see it goes, and it's a little less hydrophobic, but then it's really hydrophobic again. If we go back to the actual structure, you'll see where that is. And so here is what I was talking about before. So they have taken all of the charged, so you can see the hydrophobic are, white, silver, or gray color. All of the charged amino acids are the dark color, and then tryptophan and tyrosine, which are the aromatics, yet they also have at least one polar atom. Tryptophan is still pretty hydrophobic, but it does have a nitrogen, in it. And so if you look at these, it doesn't matter which one we pick. This is the potassium channel, which is, we're going to go into greater detail with it. It's really cool. To me, it kind of looks like reminds me of a nutcracker, except it's bidirectional. But we see it's highly charged on the outside and inside. And then right there at that cap, right at the exact level, we have those tryptophans and tyrosines that kind of help hold it, but kind of hold it in place. And then it's really hydrophobic. If you look at multiporin, you see a very similar situation. We actually do have some charges, but they're charges with respect to in the same general area to help hold it together. But you see the same principle where you get this hydrophilic region, this aromatic, like ring or streak right there at the head group area, the lipids, and then the hydrophobic interior. And so it's just really, really cool. And then once again, you can look at the hydropathy index and hydropathy plots to see and to correlate the fact that this is also true, that you have these hydrophobics versus the hydrophilic regions. So, are there any questions?